Nancy Baker here. Today's topic is mesh mining. I am here on the abstract sifter on the main sheet. I've just run a query. The query was cornea, major, and chemicals. So I'm running this query because let's say that I'm interested in chemicals that both treat corneal diseases, or maybe even I'm interested in chemicals that cause problems to the cornea. So I'm looking for those relationships. I've run this query. It's giving me about 2,000, yes, over 2,600 articles, and there they are on the main sheet. So mesh binding means that the abstract sifter is going to go to each one of these citations and get the mesh substance terms off of those citations and then collect them and let me look at them. So let's talk a little bit about what the mesh vocabulary is. MESH stands for medical subject heading, and I'm going to go to one of these articles here that I've already picked out and show you how to look at those on the PubMed website. So for this particular article, I'm going to scroll down, and here is the hyperlink to the mesh terms. And here are the mesh terms, and you can see that they're partly anatomy terms, they're species, they're also chemical terms. This asterisk means that this chemical is a major topic of this article and that it's about the therapeutic use of this chemical. The substances sort of breaks out the chemicals from this mesh term list, and this substance list is what we're going to extract from all the 2,000 plus articles on the main sheet. To do that, we're going to be able to get a great overview of the chemicals associated with the cornea, and then look at whether they seem to treat diseases with the having to do with the cornea or cause problems, toxicities to the cornea. So let's figure out how to do this. And of course, this is just an example. You can use this functionality for any number of different corpora. So I've got the main sheet. I'm going to click on More Things, and I'm going to extract mesh substances. And I'm going for major topics only because I just want to start with that. I could spread out later. And now what the abstract sifter is doing is going to every one of these 2,000 plus PubMed citations and looking for the annotated substances. It's going to collect them, count them, and place them here, 1,389 of them, on this sheet called the MeSHMine sheet. So let's take a look at what was found. Here in this column A are the substances, and they are hyperlinked to the mesh browser, and I'll show you that in a second. But you can see there are all sorts of different names. There's very broad names, family names, and then there's specific acids, very broad, very specific names too, like chlorhexidine gluconate. And so we could sort these alphabetically, we could sort them any number of ways. But what I think I'm going to do here, first of all, is clean off the family names, which are really too broad to be useful for my particular purposes. So now we just have specific chemicals here. If you, this first, column C here has the article count. And that means that, say, collagen was a major topic in 94 of these articles. Mustard gas was a major topic in 27 of these articles, and in 26 of them it was described as being toxic. You can also see that some of these chemicals are described as in relationship to their therapeutic use. And then we can take a look at a couple of those in a second. Over here are some flags, or 0 and empty here. So it's 1 if the flag is on. And these are categories, or these are categories, branches from the mesh tree for substances. Let's look and see how that works. Let's look at mustard gas. I'm going to click on that, and I'm going to be taken to the National Library of Medicine mesh browser and the record for mustard gas. And you can see here the tree number starts with DO2. Here it is, and it's under organic chemicals, which is DO2. If I go to the tree view to look at all the whole tree, you can see that these are all the categories, branches of trees under chemicals and drugs. And that's what we have reproduced here, so you will know if it's some chemical you've never heard of, something about it by looking at these ones, these flags. Okay, so let's dig in. So let's go ahead and click on this mustard gas 27 here. All that does is write the word mustard gas to B3, this sifter term here. And of course, sifting, anything you write in B3, C3, or D3, the abstract sifters automatically count the occurrences of the string of characters here in all of these titles, abstracts, and keywords. And this one has the most occurrences of mustard gas. And let's actually then put cornea here and double click and see what we can find here. So mustard gas keratitis, 38-year-old man, corneal topography. So you can sort of skim really quickly and see the relationship between mustard gas and a cornea. Also, if you're looking at finding chemicals, that treat diseases having to do with the cornea, you can sort of therapeutic use, and let's go down to whatever this is, netasterdil, and double click there, and it'll write it here. Reversible corneal endothelial abnormalities with netasterdil. Okay, so there must have been some AE in tox. Yes, so let's go here and look at this one, see if it's a treatment for some corneal disease. Well, okay, I don't really understand what all those things say, but alanine edetic acid, let's click here, disruption of urine eye dry. Okay, it's abrogated by methylprednisolone. Some of these are actual treatments for some corneal diseases. So that's how you can then sort of explore the space and look for chemicals that you might want to study further, either for maybe their toxicity or maybe for their therapeutic use. And of course, you can take these chemicals. Let's go ahead and sort by article count. You can take these chemicals and copy them and paste them over here in the landscape sheet. I just did this with some genes and proteins, and go out and do some more queries to get more information about their relationship to the cornea or eye diseases. One of the important flags here is this protein, gene, or amino acid flag. If I sort by that, I get the genes and proteins that have been annotated in this corpus of articles about cornea. So if you do the right query and you come and look at the genes and proteins, you can get a picture of the proteins, genes that are players behind a certain part of anatomy or a certain process in biology or disease state. And that's an introduction to the MeSHMine capability in the abstract sifter. Thank you.